बोला
ते थोडंसं जवळ घ्यायचं आहे. एक मिनिट, मला दिसणार नाही. तिकडे दिसतंय का? दिसतंय का? आवाज येतोय? करू. Good morning all. Welcome to another session of YouTube live stream of Standard 10 Science and Technology Part 2. I'm Mrs. Priya Mohorkar from SV Union High School and Junior College, Pune, Maharashtra, India. Welcome you all to this new session of Chapter 8, Cell Biology and Biotechnology. Well, we all are living in this world nowadays, consuming the yields of the products of this field only, and we are going to use this field and explore it, exploit it, as well as make it useful for the whole of mankind. Let us first recall the basics for this chapter. Yes, so you all know that all the living organisms in this world, whether it is a unicellular, a single-celled organism, or a multicellular complex organisms, all these are made up of basic building blocks called cells, right? You have learnt in your earlier classes, a cell is a structural and functional unit of living beings, as it forms the structure and it is going to function for the whole organism. That is why it is the basic structural and functional unit of life itself, right? Cells that have the same structure and perform the same functions is generally called as a tissue. The cells actually, you all know, would have formed with a very simple structure organized into prokaryotic and very basic functions, like the cells showing the prokaryotic organization as in the case of bacterial cells, right? Slowly, there were some certain modifications, certain advancements in the structure as well as functions, and the cells started showing changes, modifications, and they started having the eukaryotic organization, wherein the nucleus played a very important role along with all the other cell organelles, may it be plant cells or animal cells, right? So the cells would have come together slowly and carried out different functions. They started dividing the labor, they started dividing their functions amongst themselves, and would have started modifications as well as changes according to the functions. Thus, the cells that had the same structure and which performed the same functions started working together. Now, this group of cells is called as a tissue now. Very well, the different tissues in the same manner would have together formed an organ, right? And an organ, different organs would have arranged together as an organ system. Organ systems like the digestive system, the respiratory system, the nervous system, all these different types of organ systems would have been formed by the arrangement of different organs in them. All the organ systems together form the whole organism, which is a complex multicellular organism and performs various different types of functions. Let us see how did this organization of organism has formed the basic of the hierarchy, wherein a single cell, the individual cell would have slowly formed tissues when it started grouping together, performing the same kind of function. So this tissue would have slowly differentiated into different types of other tissues and would have later on formed organs, different organs, like here stomach, liver. Of course, you have different other organs like brain, kidneys, you have heart, isn't it? So different large organs would have been formed. Now, when these large organs would have arranged themselves, we call that as an organ system. For example, the digestive system, okay, that shows different organs that are grouped together and performing, of course, parts of different functions carried out by each part. Still they are the part of the same system, that is the digestive system. The organ systems, different organ systems later together would have formed the organism, right? So this is how the hierarchy of organization of organism is set up, right? So let us talk about this organism. In an organism, we know the organism is made up of basic building blocks called cells, isn't it? Now, some of these cells are said to be totipotent. Now, what is this totipotent? Actually, toti refers to total and potent refers to potential or the capacity, okay? Now, in an organism, some of these cells are totipotent. They have the capacity, total potential in them to produce a whole organism. So few cells have this potential to produce whole organism if at all they are grown, they are nurtured, they are cultured in a proper environment, okay? A proper environment may be controlled environment, which we can call it as a lab, okay? Right. Now, this property of the cells, that is the totipotent property of the cells, actually has led to the rise of a new branch of biology itself, which is known as biotechnology. So initially we all were in a state where we only had studied the structure of a cell, a prokaryotic cell, a eukaryotic cell, their organization, their functions in different conditions. What are those functions? How complex those functions are, and everything we were studying under what is called as cell biology or cytology, wherein we learn about the structure and function of cells, right? Whereas because of this property called totipotent property of few of the cells, a new branch of biology slowly started developing, which we call it as biotechnology. With the help of this technology, it has become possible to produce improved varieties of plants as well as animals for human welfare. So whatever we study, whatever we try to use, whether it is in various fields of science, is for human welfare, remember, okay? Human welfare only, and the welfare of the mankind only. Right, so let us talk about what is exactly cell biology. Now, cell biology is the branch of biology concerned with the study of structure and various functions of the cell, okay? So the branch of biology concerned with study of the structure and function of the cell is called as cell biology or cytology. Cytos refers to cell and logos refers to study or the discourse. of that field. Now, in recent times, there are various revolutionary changes in this field, in the field of human health actually, due to this field, this branch called as a cell biology. The study of the structure and functions of cells are generally are actually being studied in various institutes. Even now, research is going on the structure and function of the cells. But in India, there are two institutes which have dedicated which are dedicated to the study of cells, and that is National Center for Cell Science at Pune, Maharashtra, and the other institute is InSTEM at Bengaluru. All these both institutes are very good where the cell science or the structure and function of science of the cell is being studied in detail. You can visit these institutes for better understanding. Let us see what is biotechnology. Now, biotechnology is a new field for you all students, where you can explore, you can do lots of research, the techniques of bringing about improvements in living organisms by artificial genetic changes. So changes in the genes, in the DNA, can be done by artificial techniques, and producing hybrids by hybridization for the welfare of human beings. Together is called and studied under biotechnology. Biotechnology actually has two different methods. Okay, the basic method is tissue culture, and the other advanced method, technique, where different techniques are used, is the genetic engineering. Let us talk about who discovered the basics of biotechnology, that lies actually in the totipotency, the capacity of cells and tissues to produce the whole organism. Let us see the discovery of totipotency of cells and tissues. It was Frederick Campion Steward, who was a British botanist. He actually showed that cells and tissues can be grown ex vivo. Ex refers to exterior, vivo refers to the living system. So exterior to a living system, the cells can be and the tissues can be grown in controlled conditions. So he actually performed experiments on carrot root. He isolated the cells from carrot root and cultured them, grew them on a nutrient medium. He also proved that each cell has the ability to produce a whole new plant. As he was working on carrot root, the whole carrot plant was developed by him. So let us see what exactly this tissue culture technique has in store for us. Ex vivo growth of cells or tissues in an aseptic and nutrient-rich medium in controlled conditions is called as tissue culture. Now, ex vivo, I told you already, it is growing cells in exterior medium, where there is no living system or living medium over there. So ex vivo growth of cells can be done in aseptic conditions. Now, what is aseptic condition? Aseptic is germ-free condition, where there are no germs, no pathogens that may infect or affect the growth of the cells. And the cells are grown in a nutrient-rich medium containing water mixed with agar and all the other minerals, nutrients, along with the growth hormones and vitamins. So whatever is necessary for growth of the cells are provided with all that those nutrients in the rich medium, and they are grown in controlled conditions, that is lab conditions, where the physical conditions can be controlled, like temperature can be controlled under optimum temperature, such as the optimum temperature for growth of most of the cells, plant cells as well as animal cells, is generally 25 degrees Celsius, which is considered as the optimum temperature, or the temperature should be optimized for certain cells wherever necessary. So so not only temperature, light can be controlled, moisture content, humidity can be controlled. So various physical conditions can be controlled in a laboratory, and that is where the ex vivo growth of cells or tissues is possible under this technique called tissue culture. So tissue culture is that technique in which ex vivo growth of cells or tissues in an aseptic and nutrient-rich medium in controlled conditions is possible. Now, the tissue culture technique was possible only due to the totipotent or pluripotent capacity of the plant or animal cells. So if it is going to be a plant cell, then the capacity is called as totipotent capacity. Animal cells being pluripotent, that is the capacity of animal cells to grow in controlled conditions. With the help of this tissue culture technique, a complete organism can be developed from a single cell or a group of cells, which we call it as tissue. So you can see here, if at all a leaf is taken, a plant part, which is a vegetative plant part, a small part of this leaf can be removed and used as an explant, can be grown in controlled conditions. Small explants or small parts of this leaf can be placed in the nutrient medium, the agar medium, which is of course placed in a kept in a aseptic and controlled conditions in the lab, providing the nutrient-rich medium for growth of these cells. Now, new cells develop as a result of cell divisions, and a group of cells called callus is, you can see the callus grown in this petri dish, which is generally used in tissue culture techniques. Not only that, even in microbiological techniques, petri plates are being used for the growth and culture of bacteria and fungi. Anyway, such petri plates are being used to grow the callus when the explants are grown, inoculated and grown in these petri dishes. You can see the group of cells. These cells are just they just multiply and grow into what are called as callus. Okay, now these calluses can be separated and can be inoculated in different petri plates or bottles, or even conical flasks with agar medium, nutrient rich agar medium, that is provided with growth hormones. Now, these growth hormones decide the growth of the vegetative parts, like the roots, the stem, the leaves. You can see a single plant which is being propagating in the petri dish from the callus, actually, a small part of the tissue of callus. So this is how multiplication of various various plants can be done and is possible nowadays. Of course, we all are, I told you, we are living in this technological world. Of course, we are growing on, we are surviving on this biotechnological field, right? So this is how generally a plant can be, a small part of the plant, a group of cells, a tissue can be multiplied, okay, by the technique of tissue culture. Tissue culture in plants, let us see. Now, we just saw that a leaf can be used, a leaf part can be used, a root part can be used, parts of the stem or the growing tips, that is, which contains the meristematic tissues. These tissues are all termed as the pluripotent tissue. They are totipotent tissues, as they have the capacity to grow into or develop into any type of further tissue. So generally, you all know that in plants there are two different types of tissues. You have learned in the earlier classes. They are the simple tissues and the complex tissues. The simple tissues basically are parenchyma, collenchyma, and sclerenchyma tissues, whereas which are the simple permanent tissues, and the complex permanent tissues are the xylem and phloem, which contain each one containing 4 different types of tissues that are actually involved in transportation, or they are conduction of water and minerals. Well, the other type of tissues are the meristematic tissues. These meristematic tissues are also totipotent cells, which can develop into any type of cells. They are generally found in different parts like the stem tip, the root tips, the leaf tips as growth tips, and the flowers, the buds, okay, the growing buds. So these are the places where the meristematic tissues in a plant can be found. But not only them, even the simple permanent tissues can be used, because they also have the basic totipotent capacity to grow and develop into a new organism. Now, if you see in this picture, you can take a tissue sample scraped from a parent plant, from the leaf of a parent plant. The leaf part is the petiole part. A small chips of the petiole can be taken as explants, and these explants are transferred into the agar growth medium containing nutrients as well as growth hormones, auxins, in combination with cytokinins and gibberellins. These explants grow into calyces, and calyces can be further developed into tiny plantlets by introducing the growth hormones in the agar medium. Now, the next stage is the tiny plantlets are slowly planted into the compost for under hardening process, and once this process, the plantlets complete this process, they can be taken into the field in natural environment. That comes under the process called secondary hardening. Now let us just have a look at various processes in tissue culture. If you see that a source plant is taken, any part of the plant, the growing tips of the plant can also be taken as a source, and the explants maybe from, in this process, the explant is taken from the bulb corm of the banana plant here shown. Now, these explants are actually, they are transferred into agar growth medium for primary treatment, that is formation of callus. You can see very well that the callus is grown in the bottled, in the agar medium inside this bottle. Not only that, slowly the cells, the callus undergoes multiplication and develops a shoot system, right, because of the addition of growth hormones in it. Now, not only that, the development of roots also take place. You can see shooting and rooting taking place during the multiplication process in the process of tissue culture. Now, these small plantlets are slowly taken up for primary hardening, and they are in course of time they can withstand the soil conditions outside the lab, outside the controlled conditions in the natural environment, and slowly transfer to pots and later on to the fields under secondary hardening, wherein the plants which were grown in controlled conditions slowly can get acclimatized to the natural conditions. This is how the processes in tissue culture leads to the multiplication of the required plant. Now, this process is actually a very, very important process, basic process, wherein you can see here the primary, the plantlets of the banana plant um, has been allowed to acclimatize in a greenhouse. You can very well see how the plantlets have started growing and getting accustomed to the environment, natural environment, and later on these plantlets can be used for farming. Okay, the plantlets of banana are being used for farming. Here you can see the plants that are multiplied in the lab conditions are now growing very well in the natural environment conditions, right? That is the power of tissue culture. That is the power of the totipotent capacity of the cells. That is the power of life, students. So let us talk about the other method by which biotechnology is nowadays ruling the world. That is making changes in the genes, engineering methods being applied to the genes, to the DNA of the cells, which is present inside the nucleus of the cells. And that is what the whole world is consuming, the yields of this particular method of biotechnology. So genetic engineering is actually the deliberate modification of the characteristics of an organism by manipulating its genetic material. The genetic material is that DNA in general. So this method wherein man has tried to manipulate, to change, to modify the structure of the gene, the structure of the DNA, so as to produce desired characteristics in organisms. May it be plant, may it be animals, may it be microorganisms, right? So genetic engineering has paved way to production of the genetically modified crops. Here you can see the genetically modified crops and other plants which are produced by introducing changes in the DNA of natural crops and of course other plants also. These GM or plants or the genetically modified plants exhibit the desired characteristics, like characteristics to withstand environmental stress, insect resistant, pest resistant, chemicals resistant, drought resistant, weedicide resistant varieties, and many more different types of varieties. Hybrids are being produced. Better quality seeds are being produced. High yielding varieties of the genetically modified crops like banana, maize, rice, cotton, tomato, potato, etc. are produced with the desired characteristics. May it be seedless varieties, may it be variety with large yield, not only yield, may it be yielding large and hybrid varieties with larger size and more taste, right? All these things, and with more amount of seeds. So all these better quality crops and plants is being produced because of genetic engineering process of biotechnology. Another very important and very illustrative, very useful are those for the mankind, useful for the whole of mankind, are the stem cells. Now, what are these stem cells? These are special types of undifferentiated cells, students. And these cells are present in the body of an organism itself. An animal that gives rise to all other types of cells. So till now we were talking about the totipotent capacity of the cells, basically in plants, right, where tissue culture is just flourishing to multiply the different types of plants, right? Now these cells are also these kind of cells having the same kind of capacity, potential to produce the whole organism is also found in animals, in human beings, right? Now what are these cells? These are special kind of undifferentiated cells, cells which are not undergone any kind of differentiation according to their functions. And these types of cells actually have the capacity to produce the whole new organism, right, on their own. So that these cells are termed as the stem cells. Got it? Now, but where and how do these stem cells are produced in the body? Let me tell you, after the union of male and female gametes, where we call that the zygote is formed, a new life emerges out of the haploid cells during the sexual reproduction. Now this zygote that is formed divides and redivides to form a new organism, into a fetus, into a small child, isn't it? Of course, it is the next generation of the parents. Now, when this particular zygote is formed and it divides and redivides by mitotic process, it produces a group of cells, right, during the early stages of development. We call this resultant product as the embryo. The embryo has a mass of cells, right? A mass of cells wherein the cells are grouped, they are not differentiated. They are a group of cells which are not differentiated into any other type. Okay, so each one has the capacity to develop into any other type of cell. Such cells, these cells which are found in this embryo, the embryonic cells, are called as the stem cells. These stem cells has the capacity to develop into any type of cell by the process of differentiation. Okay, now differentiation is changes that occurs in the cell to perform a particular function in a particular organ, in a particular position that it is present in. Now the stem cells are thus are called as the pluripotent because they have the capacity to grow into a new organism. Pluri refers to plural, many different ways in which the cell can show the capacity to produce a new organism. So these stem cells are said to be pluripotent cells. Now let us talk about where these stem cells are located in man. Location of stem cells in man. Let us just take a look at this. Now, in human beings, in man, these stem cells are generally located in different places, like the skin, the epidermis, that is the skin, the liver, the adipose tissue, that is the fat storing tissues just under the skin, and of course on the surface of every large organs. Not only those, the skeletal muscle cells. Okay, they are also found in brain, the glial cells, and the connective fat cells, the glial cells, which are present in the brain. The heart, the inner walls of the heart, of course, contain these stem cells. The bone marrow, the red bone marrow, which actually produces the blood cells. So even the blood cells are the stem cells, but only when they are located in the bone marrow. All these are the stem cells in man. Now, the location still to be very particular. Let us see the stem cells in man, where exactly, from where they can be collected. in umbilical cord by which the fetus is joined to the uterus of the mother, the stem cells are actually present. Okay, the stem cells can be also collected during the immediately after the birth of the child. When the umbilical cord is cut, the umbilical blood cells as well as the umbilical cord tissues can be utilized and stored further because they are the stem cells, right? The location, another location of the stem cells is the blastocyst stage of the embryonic of the embryo during the process of embryonic development of fetus, wherein you can see the, of course, as I told you earlier, fertilized egg. When it starts dividing and redividing, the zygote starts dividing and redividing, slowly starts growing into 2-cell, 4-celled, 8-cell, 16-cell stage of embryo. Later on actually grows into a mass of cells called morula. Now this morula, the cells which are irregularly arranged in a morula slowly starts arranging itself in another structure called blastocyst or blastula. Now this blastula contains the cells that are the stem cells. These stem cells actually can be called as the embryonic stem cells, which can be collected and of course used for the culture of organs and the whole organism. Their pluripotent capacity can be explored. Another place where the stem cells are found are the red bone marrow of adult human beings, right? The bone marrow cells, which are the hematopoietic cells, stem cells, can of course be utilized to produce red blood cells, white blood cells, as well as blood platelets. Not only that, the whole organs can also be developed. The next place where the stem cells can be located is the adipose connective tissue, where the fat cells, the adipose stem cells, can be used to produce different other types of cells like the chondrocyte, the osteocyte, the bone cells, the adipocyte, the fat cells, the hepatocyte, that is the liver cells, the tenocyte, the neurons, that is the brain cells, the myocyte, that is the muscle cells. So different kinds of cells can be produced from the adipose connective tissue, right? Which are of course place of the stem cells, location of the stem cells. The adipose tissue, you all know, are generally found under the skin and on the surface of large organs, and they help in they connect various organs to each other inside the human body. So let us see how these stem cells are preserved. The stem cell samples are carefully collected from these sources like the cord blood, the umbilical cord blood, the red bone marrow or embryo, from the blastocyst mainly, right? And these collected samples are kept in small sterile vials, small bottles, which are actually kept in liquid nitrogen at a particular temperature of minus 135 degrees to minus 195 degrees Celsius. So these stem cells can be preserved in this manner, but they are preserved mostly in various institutes wherein they are actually the stem cell banks. Okay, cell banks, like you have the gene banks, the seed banks nowadays. The same way, stem cells bank preserve these stem cells very carefully later on, which can be used whenever necessary by human beings, by man. Let us see how the stem cells research actually started and carried out. Actually, it is a revolutionary event after the cloning process in 1996, where you all have heard about the Dolly sheep that was produced by the method of cloning. Now, this technique, the technique of stem cells research technique has brought about fundamental changes in the field of medical sciences. Okay, now this is because of different types of stem cells. There are basically two types of stem cells that we are going to see now. One is the embryonic stem cells and the other is the adult stem cells. What are these embryonic stem cells? Let us just have a look. The embryonic stem cells are actually the cells which are formed after the when the zygote formed by the fertilization of the union of the fusion of male and female gametes divides and redivides. The zygote divides and redivides to form a group of cells called as the morula, right? By simple mitotic division, it forms the zygote is converted into embryo. We term that as the morula. Now, generally, after this morula stage, it gets the cell starts arranging itself in layers. Okay, the stage being called as the blastula or the blastocyst. Now, the cell differentiation generally starts from the 14th day of conception, that is formation of zygote. Now, cells of different organs, right, slowly starts forming after the 14th day, okay, of conception. Now, what are the different types of organs and cells that they are formed? Like they get differentiated into osteocytes, that is the bone cells, the hepatocytes, like the liver cells, the neurons, the adipose tissues, the connectives, and different types of epithelial tissues by the process of differentiation, redifferentiation and dedifferentiation. Okay, so these embryonic cells before differentiation are actually called as the embryonic stem cells. Now these embryonic stem cells are the primary type of undifferentiated cells. These cells have the ability, the capability of self-multiplying. That is, they can undergo cell division, mitotic cell division on their own. Now, these cells, embryonic stem cells, are actually considered as the parent cells of all the other types of human cells. The property of stems, this property of developing and differentiating into other types of cells is called as the property, the pluripotency or the pluripotent property of the stem cells, right? There are actually nearly 220 different types of cells in human body that are formed from these embryonic stem cells. Now, it has been found that if these stem cells are being collected early, well before the beginning of the differentiation on the 14th day, that is during the 5th to 7th day, and if they are cultured with some biochemical stimulus in the laboratory, then as per the stimulus that is being used, they can transform themselves into the desired types of cells, whereby the tissues and of course and finally into organs, which is desired organ. Now, thus their pluripotency can be explored to further regenerate the organs necessary for transplant. Now, the embryonic stem cells, if you see, they can be collected between the 5th and the 7th day of development, early development of embryogenesis. Now, you can see here if the cells being collected from a 4-celled embryo, not only that, from the morula stage, the cells can be collected and they can be cultured in a nutrient agar medium, so where the undifferentiated stem cells can slowly get differentiated into different types of tissues like the cardiomyocytes, that is the heart cells, the neurons, which are the brain cells, the liver cells, or any other desired type of cells can be grown in the controlled conditions. Now, a stem cell can be used, the pluripotency of the stem cell can be used to develop into different other types of cells, which the stem cell is being collected from the blastocyst of the embryo. The embryonic stem cells being the parent cell for different types of cells, like the epithelial cells, the chondrocytes, the red blood cells, the fat cells, the enterocytes, the cardiac cells, the muscle cells, the neurons, and any other type. Okay, the second type of stem cells are the adult stem cells. These stem cells can be obtained from an adult person too. Okay, now there are three main sources of stem cells in the body of an adult person. Now, which are those sources? Which are those locations? Let us just see. The first is the bone marrow, the red bone marrow mainly. Okay, there are two bone marrows. One is the red, the other one is the yellow. Now, from the red bone marrow, we can extract the adult stem cells. Second is the blood cells from the blood. Okay, and the third, the adipose connective tissue. Not only that, from the cord blood. That is the just after delivery, when the cord between the mother and the child is being cut, the umbilical cord that is being cut allows oozing out of cord blood. Now, immediately after birth, if that cord blood is collected, then it contains the adult stem cells. Right? So here you can see the blood cells that are actually the mesenchymal stem cells that can be used to culture various other different types of cells. The red bone marrow containing the stem cell can be utilized to produce different types of blood cells, right? The adipose connective tissue cells can be utilized as adult stem cells to grow different other types of tissues, desired tissue, and can be converted into various organs. So adult stem cells generally can be not only from these four different locations, but they can be extracted from many other basic locations like the skin, the digestive system, the cornea, the umbilical cord, the spinal cord, the liver, the fat cells, the bone marrow, the dental pulp, the pancreas, the brain, the peripheral blood vessels, the retina of the eye, and the skeletal muscles. So there are many different places from where the adult stem cells can be extracted. Now let us see the uses of stem cells. The stem cells are generally used in the regenerative therapy, like the cell therapy, and to produce the blood cells. The cell therapy, actually the stem cells are used to replace the dead cells in case of conditions like diabetes, myocardial infarction, Alzheimer's disease, Parkinson's disease, etc. Now here is a picture of myocardial infarction, where you can see the infarction, the damaged tissue on the heart, on the surface of the heart, on the muscles, muscular wall of the heart. Now the stem cells inside the heart, inside the walls of the heart, can be utilized to generate or to produce the cells, the heart muscle cells, and they can be repaired. You can see the other materials and scaffold can be is released, and that part is recovered. So myocardial regenerative treatment can be done using cell therapy. Not only that, to produce blood cells, which is required, blood is generally required in conditions like anemia, thalassemia, and leukemia, wherein the stem cells can be utilized to produce the blood cells. Now another use is to in organ transplantation. In case of failure of any organ like kidney, liver, etc., they can be produced with the help of stem cells. Like here you can see organ, that is the ear pinna, is being generated, not only regenerated, but it is also transplanted successfully. Other uses, you can see how the stem cell can be actually utilized to grow various organs like brain, the intestines, the liver, heart, and the bones through differentiation of the stem cells into various types of tissues. Not only that, various organs in the human body either become less efficient or completely functionless due to various reasons like aging, infections, accidents, disorders, etc. Life of such persons become difficult, or sometimes fatality occurs in such conditions. However, if a person gets the necessary organ under such conditions, then his or her life can be saved by organ transplantation. The availability of a donor is important requirement in organ transplantation. For example, each person has a pair of kidneys and excretion can occur with the help of single kidney. The person can donate one of these kidneys so that the other person, the patient who is devoid of kidney or devoid of a functional kidney can utilize it. The skin from some parts of our body can also be donated. But what care should we take during organ donation and transplantation? Let us see. Various factors like age, diseases, blood group compatibility, etc. of the donor and the recipient need to be paid attention during organ donation and organ transplantation. All the organs cannot be donated during lifetime. Okay, organs like liver, eyes, heart can be donated only after death. That is called as posthumous donation of organs and the body. In case of organ and the body donation, human bodies are, you all know, generally disposed of after death as per traditional customs. Due to the progress in science, it has been realized that many organs remain for certain period, live for certain period of time even after death. Concept like organ donation and body donation can be used to save the lives of the needful persons so that their life would become comfortable. People can volunteer such donations even after death. Organ and body donation can be done so that life of many people can be saved. Organs like liver, kidney, eyes, skin, heart valves, etc. can be donated after death so that people are benefited. Body can be made available for even research and medical studies. Many government and social organizations are working for this and towards the increasing the awareness of body donation. You can also visit websites regarding organ donation, that is www.organindia.org. And I take this opportunity to request each and every individual to donate your organs and your body for the benefit of mankind. May it be after death, but still it can be done so. So please donate organs for the benefit of mankind. Thank you all of you for joining me this session. Let us meet again in the next session of the same chapter. Thank you all of you. Have a very good day and good life. Thank you.